Thank you all for being here. Thank you again for saying such nice things about me. So obviously, I'm going to talk to you a little bit about gene transcription and about my time in graduate school. And particularly, I'm not going to tell you very much about my data. Instead, I'm going to talk some more about some of the questions that I've asked to get me to this point, and maybe I can answer some of yours in the process. So here's sort of the standard training period for biological scientists. We have this basically 4 years of undergrad and 5 or 6 years of graduate school. I'm a fourth year PhD student, so I can look back on a lot of this at this point. And as was mentioned, I did my undergraduate degree at Case Western Reserve University in Cleveland, Ohio. I'm from Ohio. That sounds far away to you. I promise that California sounded farther away to me. I had never been to California until my interview at Scripps Research, actually. And I really loved it at Case, even though it did look like this more of the time. I literally walked to my 8 a.m. biochemistry class through 2 feet of snow at 0 degrees Fahrenheit 3 days a week. And by the next time I tell that story, it probably will have been uphill both ways as well. So obviously now I'm in beautiful La Jolla at Scripps Research. And my notes have turned off here. Come back. So when we look at this sort of timeline, my path seems very straightforward, but when I look back at it, I see a lot of coincidences. So I have this little timeline here. It's going to be over on the left. So when I was an undergrad, I actually started out as an undecided major. You might think I remained undecided since I ended up with 3, but I didn't always want to be a scientist. And so I actually took freshman biology my sophomore year of college. And that freshman biology class is really where I started to get excited about biology. I'm not really here to cure disease. That's a very wonderful goal, but it's not what I'm excited about. So instead, I actually got excited when I saw this slide in freshman bio class. I know it's really inspiring looking. So this is a codon table. This is the code of DNA and RNA that allows cells during gene expression to produce useful proteins from their genes. And this got put up in that freshman bio class, and I was like, there's the meaning of life. It's all on this one slide. Because every organism on Earth, from bacteria, trees, giraffes, what have you, uses this same code. And what I think is really beautiful about it is the fact that it's not like cells look up this table when they're producing proteins. This is a chemical code, and it's so well organized that it proceeds naturally without a cell needing to know the bigger picture that it's involved in. So that's the kind of problem that I really like. I really like understanding how these complex chemicals lead to functional molecules, produce cellular organization, and lead to complex organisms like you and I. To put it in another way, would you mind bringing me my water? Thank you. I like to look right underneath the hood of what's alive, this stuff. Does this pointer work? I like to look underneath the hood of the stuff on the left that's alive at these functional molecules that are not alive and ask what about them it is that lets them to contribute to something that's alive. And so that's what I think about biophysics. And we're still at the beginning of my undergraduate career here. I didn't know the word biophysics, but it's how biological molecules move and interact. And even though I didn't know this word, this is what I was getting really excited about. So in biophysics, we use the techniques and the concerns at the molecular level, so your chemistry, your thermodynamics, your kinetics, and we take those and point them at biological systems to try to understand them better. So I declared a biochemistry major. And to my great chagrin, you were required to do research in order to graduate as a biochemistry major. I was quite upset about this, but I didn't have a choice. So I typed up some emails to a couple professors in my department I just kind of like looked at their lab websites, decided who I thought sounded the coolest, sent out some emails. 2 of them answered me, and I picked one. And here we are. And the question that I had in the course of that was, which science sounds the coolest? And in retrospect now, I think that was the wrong way to go about this. First of all, because not everybody keeps their lab website up to date. But second of all, because there are so many great scientific problems out there. And I was worried that I was picking the one that I needed to stick with for my entire career. And that's not the case. You're going to have a chance to change topics as you enter graduate school, if that's what you're interested in, as you leave graduate school, sometimes in the middle of graduate school. Like, you're never going to get stuck in one career topic. So keeping in mind that there are many, many wonderful problems out there to work on, instead I think you should be asking, who's going to value teaching you? Even if that person isn't necessarily the professor in the lab. I actually was mentored almost exclusively by a staff scientist in the lab that I worked in who taught me everything that I learned in my undergraduate. And beyond, do they value mentorship, you also need them to have a mentorship style that's going to match your learning style. So whether you would prefer someone to be a little more hands-on in teaching you, or if you want to be left alone to be productive, that matters. And I just want to put a little plug in here. There's this article, How to Pick a Graduate Advisor by Ben Barris. It's in the journal Neuron. You can find it by Googling it, and I found it really helpful. Both, I think it'll be useful if you want to go to graduate school or if you're just looking for an advisor to work for as an undergraduate. So I was finishing up my bachelor's degree, and among my peers, as far as I could tell, you could get a biochemistry degree and either go to medical school or quit. I had lots of friends be like, okay, I'm done, I'm gonna go be a lawyer now. And I was like, but I like biochemistry, like, that's why I'm here. So the other path here, of course, was I had all these professors who had biochemistry degrees. And I was like, okay, well, that's clearly what you do when you like biochemistry. I'll go be a professor. Perfect. But what I left out when I mentioned that I picked my lab based on just which description of the scientists sound the coolest, is that there were no other students in my lab. I did not know any graduate students. I didn't know any postdocs. I didn't even know any other undergraduates who were interested in doing science in my field. So there was this huge question mark in between where I was and how you become a professor. For the record, there's many other things you can do with a graduate degree than become a professor, but we're talking about, just about to graduate college, Emily, so I didn't know that yet. But I didn't really recognize this box of question marks here, how much of this that I didn't know until I got to graduate school. And suddenly I was hearing about how my roommate's mom was a medical doctor, and my lab mate's dad is a professor, and this friend has been doing internships in labs every year since they were in high school. And I sort of looked around and was like, I have no idea what I'm doing here. I didn't know anything about publishing papers. I didn't know anything about lab politics. I didn't know anything about getting funded. And it really started to feel like I was surrounded by people who knew way more than me about everything. And so I'm talking right now about a classic imposter syndrome diagnosis. So imposter syndrome is this idea that you doubt your own accomplishments and whether you deserve them. And it's especially common in people who are very accomplished and in people who are minorities in their fields for any reason. So I think it's important for us all to know that feeling this way is normal and also that it is a lie. So part of the reason that I was struggling with this feeling was, to be fair, that I jumped into a bit of a new field. So I'm in the Department of Structural Biology here. It's in the bottom. Structural and Computational Biology. And in structural biology, we can get information about one. These are fake proteins, is what all these squiggles are. We can get information about one molecule and use it to infer information about other molecules. And in a minute, Colby's going to tell us a really great story about structural biology. But despite being in the structural biology department, my lab specializes in unstructured biology. We work on intrinsically disordered proteins, which are basically lymph noodles, which is why I have drawn you many lymph noodles. And these intrinsically disordered proteins turn out to be really important. They play a huge part in all kinds of regulation in cells. They can fold into different states in different cellular environments, and sometimes they don't fold at all. And so when we're studying these, the problem that we run into is that each one looks a little different from the next one. We need a strategy of looking at them that allows us to differentiate among diverse populations of cells, or of molecules. And so naturally, we shoot lasers at them. And to be more precise, we put fluorescent dyes on our proteins, and then we shoot lasers at those. So one of the, as was mentioned in my introduction, I work with these fluorescence techniques. So fluorescence is when a molecule absorbs some high energy light and then releases a slightly lower energy light. And this can happen with many different colors, many different wavelengths of light. And if you get two particular fluorophores, these fluorescent molecules, with these overlapping light spectra, you can get an energy transfer between them. So you would shine light onto our green molecule over here, it's going to transfer energy to the red molecule, and then you can measure light being emitted from the red molecule. And this is called Frster resonance energy transfer, or FRET. And I think maybe some of you here have worked with FRET before, yes. What's really useful about FRET is that it's distance dependent. So when our two fluorophores are very far apart, we don't get very much signal, and when they're very close together, more of that energy is transferred, and we get a lot of signal. And here's basically what I just said, except in math instead. So these equations on the left-hand side demonstrate or describe rather, the fact that the further away we get in angstroms along the x-axis, the less energy is transferred between our two fluorophores. And so in our lab, we attach these fluorophores to proteins, and we can use FRET as a kind of molecular ruler on the angstrom scale to tell us about the conformation of the protein that we're looking at. So, if a protein is very tightly folded up, we're going to get more of this signal because our fluorophores are brought close together, as opposed to on the top protein that is more extended is going to have less of a signal from it. Now, one of the major challenges of FRET is that it can be very difficult to distinguish this top low signal that you get when you have fluorophores very far apart from species where there's just a missing fluorophore. And so to address this in the field, there's been developed alternating laser excitation. So basically we bring a second laser into this mix and we shine it at our acceptor fluorophore. So basically we have this one laser saying, hey, are you there? And we switch back and forth with this laser that's asking about the conformation of the protein, and then we get to do this. So I joined a lab that works on this in graduate school, and my PI said, hey, can you get alternating laser excitation set up in our lab? And I was like, sure, great, no problem. But you should remember that I learned to do biology that looks like this in my undergraduate. And suddenly I was being asked to do science that looked like this. And I didn't know anything about optics. I'd never worked with lasers before. Sometimes it also looked like this. This is some of the computer code that runs our lasers. Actually, this is the computer code after I cleaned it up. It was very intimidating to look at. But regardless, oh, sorry, I went the wrong way. There we go. I ended up sort of over my head on this problem that I didn't know how to solve. And the whole discussion we had earlier about imposter syndrome was not something that just went away from me at the very beginning. So around this time, around my second year of graduate school, I started really struggling with this feeling that I didn't know what I was doing. And I was very worried that the other people in my lab were going to think that I was stupid. I was very worried about being judged. And I felt so nervous about working with other members of my lab that I started working at odd hours so that no one would be around to criticize me. That is a very bad way of learning. It doesn't work. But this went on for me for a little while until a lab mate of mine confronted me about how defensive I was being and said that I shouldn't be acting like I was better than everyone else in the lab. And that was kind of my wake-up call, because it blew my mind that I could be acting through so much fear that I was going to be seen as stupid, that instead I was coming across as snooty. I was like, how could you have gotten that this wrong? But clearly, that sort of opened my eyes that I needed to fix some things. So I'm just going to give you a couple, maybe obvious bullet points so that hopefully you don't have to learn this the same way I did. First of all, you're a student, so your role is to learn. You shouldn't be expected to know everything. It's behind. Knowledge can be gained, and not having it yet is not stupidity. And finally, that you shouldn't assume you know what other people are thinking of you, because, of course, I was deeply wrong about how my lab mates felt about me. So, following this, started out as a confrontation, ended up being a really good, productive talk that I had with my lab mate. My life in the lab got a lot better because I was accepting that I didn't, in fact, have to know everything from the get-go. And after about a year of work, I did get alternating laser excitation working in our lab. So then I got to start turning this system onto a biological problem. So I told you at the very beginning that what I started out falling in love with in science was this gene expression problem of how a cell turns its genes into productive proteins. So these are the 46 human chromosomes. There's about 20,000 genes between them. And they're regulated by transcription factors. So transcription factors are these proteins that have the job of turning genes on and off. And it's their job to make sure things run smoothly, make sure none of your heart cells just decide to become kidney cells out of nowhere. And one of the outstanding problems in biophysics is how these transcription factors solve the needle in the haystack problem, because if a transcription factor has exactly one target in the entire genome, just sort of floating around in the nucleus until it bumps into the right piece of DNA is not a very effective strategy. So what my project focused on was we're working on determining whether these proteins can instead bind to DNA in the wrong place and slide along it to look for their correct binding site. And this would potentially significantly speed up a transcription factor search, but it does require a transcription factor to do many jobs. So it has to be able to move along the DNA. It has to be able to bind loosely at the incorrect DNA and then switch to a tight binding mode when it reaches the correct DNA so it doesn't just slide on past. So we're working on different types of FRET studies to be able to study this. The specific protein that I work on is called CREB. It's the cyclic AMP response element binding protein. And if you've taken a biology class, you've probably heard of cyclic AMP. It's a pretty important cellular regulator. So CREB is a fairly central protein. It's involved in memory consolidation and nutrient metabolism, and it also is involved in the infection by the human T-cell leukemia virus that causes cancer. But I already confessed to you that I am not here to solve disease. So what I really am interested in this protein is how it finds its binding site and how we can extrapolate that information to other transcription factors. And I'm actually going to skip ahead a little bit now, and I'm just going to tell you about the most exciting unsolved question in this field that keeps me coming back to it, which is the fact that, of course, this protein doesn't have eyes. It doesn't know whether its target site is in front of it or behind it. It doesn't know whether it's on the right chromosome at all. And in fact, when it's scanning, these proteins don't even move in one uniform direction. It doesn't have a memory. So it does what's called a random walk. It binds to this DNA, and it kind of scoots back and forth. And if it moves in one direction, that's coincidence. But while this would seem to be a significant challenge for a protein that needs to find its target gene this way, one possible solution for it has to do with a preference for DNA pairs. So you remember that DNA is composed of four bases that form 2 base pairs. So if our protein prefers to bind to one of those base pairs over the other, and that base pair gets more and more common getting toward the binding site, then our protein is always going to be slightly more likely to move in the direction of the binding site than away from it. Now this would be called an energetic funnel, where your protein is always being directed in the direction of its binding site. And this has been shown to be promising in computational models, and there's some preliminary evidence that they exist in bacteria. But we're still working on getting any evidence for this model in with a real transcription factor. And that for me plays right back to what I think would be the most exciting kind of evolution, is the fact that this protein, despite having no brain, can be involved in this larger organized picture. So that's what I'm working on right now. I'm going to give you a couple more pieces of school life advice before I go. I'm going to a little warm up here. So first I've got this PhD comic for you. I'll let you read it. So if you don't know about PhD comics, I encourage you to look them up, because I'm sure you need another time waster in your day. I actually, while I was applying to graduate school, read the entire archive of PhD comics at that time. They can be really fun, and like, actually when I was looking for this comic, I saw more than one that I was like, oh, thank God I'm not the only one. But if you read 3,000 of them all in a row, you'll sort of start to get the impression that graduate school is miserable, because everybody in this comic is at least a little bit miserable. And of course, I've already told you during my graduate experience, I had at least a little slice of unpleasantness in my grad school years. And I think this kind of gets treated as like a little bit of a inside joke at graduate schools, where we're all like, yeah, grad school sucks. We all hate it here. And you're going to hear that. And I want to encourage you to reject it. So if you are in graduate school, even if you're in undergrad and you are having some kind of miserable experience, I want to encourage you to look at that as a sign that there's a problem that deserves to be addressed. And not to accept that kind of jokey status quo as something that that has to be a reality for you, because no degree is worth 5 years of misery. So there's several different ways that you can make sure that you enjoy your graduate school time, because although it is hard, it shouldn't be joyless. So the first thing I've already mentioned to you is to be sure that you choose a mentor who is invested in teaching you and invested in your success. The second thing that's worked for me is making sure that I have a place where I know I belong. So this is the board of the Network for Women in Science. And for me, this is what keeps me feeling like I've done something useful, even on days when everything in the lab falls apart. If your place of belonging is your juggling club, that works just as well. It doesn't have to be something that you can put on your resume, just as long as you have something that that is always going to accept you. And the third thing is that you should be able to ask for help. So this is my email address. Hopefully I'll get to talk to you, many of you, at posters and coffee sessions today. But if you come up with a question later, you can email me. And if you forget my email address, you can email Robert to email me. We can get in touch, and I would be happy to talk with any of you. So I want to thank Scripps Research. The Denny's Lab is where I'm from. We collaborate with the Wright Lab on CREB, as well as the graduate office, and then Carrie and Robert for organizing the NSPIRE Symposium, as well as the Student Steering Committee. And finally, thank you for listening.